Today we're going to be looking at the importance of osmotic balance and regulation. Also, we're going to be looking at some differences between types of organisms such as freshwater fish and marine fish and their osmotic adaptations. And we're going to be looking at the three major forms of nitrogenous wastes. Vertebrates can be found in many different environments, and the environmental differences come with varying challenges with regards to osmotic balance. Now, the kidney is an organ that's made up of thousands of repeating units called nephrons. The nephrons are the functional unit of the kidney. And it's in these nephrons where blood pressure forces fluid in the blood out of the ball of capillaries called the glomerulus. It's found in the Bowman's capsule. And this process of forcing fluid and small molecules out of the blood results in filtration of the blood. So as fluid and small molecules and ions enter the tubule system, large molecules are retained in the blood. And then as this filtrate passes through the nephron, nutrients and ions are reabsorbed back into the blood, leaving some water behind and some metabolic wastes behind in the urine. Now, osmoregulation is the process of the maintenance of salt and water balance, or osmotic balance, across membranes within the body's fluids, whether intracellular or extracellular. And this osmotic balance involves the concentration of water, electrolytes, and non-electrolytes. An electrolyte, of course, being a solute that dissociates into ions when dissolved in water. And the primary ions that we're looking at are sodium and chloride as the major cation and anion of the extracellular fluids, but also calcium, magnesium, and potassium. Now, if you recall, osmosis is the diffusion of water across a membrane in response to osmotic pressure, which is caused by an imbalance of molecules on either side of the membrane. Now, when we looked at plants and plant cells, we saw that a normal condition for a plant cell was for it to be turgid or under pressure, and that's achieved by it being in a hypotonic solution. Whereas in an isotonic solution, it would be flaccid, and in a hypertonic solution, it would become plasmolyzed. Whereas with an animal cell, a hypotonic solution would actually cause the cell to be lysed, and an ideal solution would be isotonic. An hypertonic solution would cause shriveling in the animal cell. Now, marine invertebrates and certain vertebrates such as hagfish and cartilaginous fish such as rays and sharks are considered osmoconformers. And an osmoconformer is an organism whose extracellular fluids are isotonic to seawater. And because that's the case, there's no osmotic gradient, and therefore there's no tendency for water to leave or enter the body. And then our other invertebrates are considered osmoregulators, which means that they maintain a constant blood osmolarity despite the different concentrations that are in their environment. The body fluids of freshwater fish are hypertonic when compared to the surrounding water. And this creates two problems. Water tends to enter the body from the environment, and solutes would tend to leave the body and enter the environment. Now, freshwater fish address the issue of water tending to enter the body by not drinking water and by excreting a large volume of very dilute urine. And this urine is hypotonic to their body fluids. And to deal with the second issue, where solutes would tend to leave the body and enter the environment, they reabsorb ions across their nephron tubules of their kidneys. So that means that the ions are reabsorbed from the glomerular filtrate back into the blood. Also, they actively transport ions across their gill surfaces from the surrounding water into the blood. Now, where freshwater fish are considered hypertonic regulators, marine bony fish are hypotonic regulators. And the body fluids of these fish are hypotonic when compared to seawater. And because of this, water tends to leave their bodies by osmosis across their gills, and then of course they also lose water in their urine. And to compensate for this continuous water loss, marine fish drink large amounts of seawater. Now there's calcium and magnesium in seawater, and many of these cations that are contained in seawater that the marine fish ingests remain in the digestive tract and are eliminated through the anus. Some are absorbed though. Now to maintain the ion concentrations, most of the monovalent ions, such as sodium and chloride, are actively transported out of the blood across the gill surfaces, whereas the divalent ions like calcium and magnesium that enter the blood are secreted into the nephron tubules and then excreted in the urine. And these are the two ways that marine bony fish eliminate the ions that they get from the seawater that they drink. Now, cartilaginous fish, which are osmoconformers, retain urea. They actually reabsorb urea from the nephron tubules, which allows them to maintain a blood urea concentration that's 100 times higher than that of mammals. And this results in their blood being isotonic compared to the surrounding seawater. And this means that they do not need to drink seawater, and they also do not need to remove large amounts of ions from their bodies. Now, terrestrial vertebrates, of course, maintain a higher concentration of water than the surrounding air. Now, they do, of course, lose water by evaporation from the skin and the lungs. Additionally, through the need to expel waste, they do lose water via the urine. However, the urinary systems of these organisms have evolved to retain water. Terrestrial vertebrates are able to excrete their waste products in a small volume of water so that more water can be retained in the body. In fact, it's the loop of Henle that's only seen in mammals and birds that allows for this formation of hypertonic urine. Whereas terrestrial reptiles, on the other hand, which do not have a loop of Henle and so cannot form hypertonic urine, don't really excrete urine. Instead, they empty it into a portion of the digestive and urinary tract where additional water can be reabsorbed, and then the wastes are actually excreted with the feces. Amino acid and nucleic acid catabolism produces nitrogen-containing byproducts called nitrogenous waste that must be eliminated from the body. And since this also involves the loss of water due to the hydrophilic nature of these waste molecules, this further complicates the process of maintaining a certain osmolarity. Now, the waste that's initially formed through the catabolism of these nitrogen-containing compounds is ammonia. Via deamination of these compounds, which is the removal of an amino group that takes place typically in the liver, we end up with a buildup of ammonia, which is toxic. Most bony fish and aquatic invertebrates can actually excrete ammonia in its original form. And amphibians and mammals convert ammonia into urea, which is far less toxic. It's water soluble, it can be excreted in large amounts in the urine. Reptiles, birds, and insects convert ammonia to uric acid, which is only slightly soluble in water. It precipitates at fairly low concentrations, so that results in the elimination of uric acid using very little water. So today we looked at the importance of osmotic balance and regulation. We contrasted some different types of organisms as to how they deal with maintaining osmotic balance and also eliminating nitrogenous waste. And then we also described the three major forms of nitrogenous waste.